let's talk a little bit about stereochemistry. Now, you've all noticed, I'm sure, that, in fact, this alpha carbon is chiral, right, depending on the R group, with the exception of one amino acid. This R group is chiral, and as a result, we can have two isomers. We could have a D isomer or an L isomer, which is based on an analogy to glyceraldehyde. Or we could have R and S absolute configuration. The first definitions of stereochemistry of D and L had to do with enantiomers that were identified by none other than Louis Pasteur, I believe, dealing with glyceraldehyde. And glyceraldehyde, yes?
Is D and L similar to R and S?
Yes and no. So D and L is older nomenclature that then got revised to R and S. D and L nomenclature had to do with analogies based on glyceraldehyde that looked like this. So this was glyceraldehyde. I'm going to draw this better. CH2OH. Okay, and if we draw truly a stereochemical view of glyceraldehyde, glyceraldehyde looks like this. Glyceraldehyde. Okay, and I can draw a mirror plane. And I can draw the stereoisomer of this, right? So I have two stereoisomers. It turned out that this isomer of glyceraldehyde rotated a plane of polarized light to the left, and so this was called levorotatory, or the L form of glyceraldehyde. This rotated a plane of polarized light to the right, and dextro for right in Greek, and so this was dextrorotatory, or D-glyceraldehyde. And then what people did was, based on these configurations of glyceraldehyde, use analogies between glyceraldehyde and amino acids in order to decide whether an amino acid was or was not L or D form. If I look at the amino acids that we find in proteins, real proteins, they look like this, right? And I'm laying it out so that it looks most similar to glyceraldehyde, right? So I'm putting the carboxyl group where the aldehyde group is, I'm putting the R group where the alcohol is, the hydrogen where it belongs, and the amino group where it sits. This is what we really purify from real proteins. Does this look like the D isomer, or does it look like the L isomer? L. And as a result, we call these L amino acids. And all proteins, really important point, all proteins from all organisms on Earth are built from L amino acids. Okay? Why don't we live in a world where there are D amino acids? You know, that's like the world of antimatter. I don't know. It's like Star Trek, right? Matter meets antimatter, it all blows up. Maybe there's an alternative universe where every protein's made of D amino acids, but not the universe we live, not the Earth we live on. Here, all proteins are built of L amino acids. Do you think that all L amino acids rotate polarized light to the left? No, of course they don't. Glyceraldehyde does, but there's no correlation between when we call an amino acid an L amino acid and which way it happens to rotate a plane of polarized light. That's a misnomer based on modeling amino acids as though they were glyceraldehyde. Now, what about absolute configuration, which is the question you asked me about R and S? So as you may remember, Kahn, Ingold, and Prelog, great chemists, decided that there was enough of this L and D stuff that didn't make any sense. Let's come up with an absolute configuration. So if we put our alpha carbon here, you remember the way you do this is you put your smallest substituent in the back, right? And then you have a steering wheel of the other substituents. You have your R group sitting here. You have your amino group sitting here. You have your carboxyl group sitting here. And then you order the substituents, right, based on the rules of ordering that you should have learned in 512. And for most amino acids, this is 1, this is 2, and this is 3 in the order of preference. And that means that our steering wheel goes this way, right? And this, therefore, is this S or R? Am I S? That's right. So they're S amino acids. There's one exception. I won't tell you what it is because we're asking you that on the problem set. Okay? But most amino acids, except one, are LS amino acids in absolute configuration. Now, how were these amino acids linked together in a protein or a polypeptide? And the solution to this problem was really solved by two chemists at the same time. Emil Fischer, right, the guy who gave rise to Fischer projections and all the other aspects of organic chemistry that you know, and another guy by the name of Franz Hofmeister. Anybody heard of Hofmeister? Remember the Hofmeister series? That's him. So those two great chemists at a meeting at the Society of, I think it was the 74th meeting of the Society of German Scientists and Physicians, shows you how close medicine and science were linked, right? Sat down and came up with the way the amino acids were linked. And they recognized that, in fact, the amino acids were linked through a condensation reaction. What was happening, in fact, was that amino acid number 1, NH3 plus, plus amino acid number 2, underwent a condensation reaction. In order to form now, what kind of bond? What kind of bond is this? Peptide bond. What do we call it chemically? You're right, it is a peptide bond. In fact, it was Fischer who first coined that term, peptide bond. What kind of bond? Amide bond, right? That's why I said proteins were polyamides. It's an amide bond. Okay? Now, this is now called, we will call this in general at this point, a dipeptide. Because it's linked by a peptide bond. Okay? And now we're going to give a new name to these things. We're going to call from now on, we're not going to call this an amino acid anymore, but we're going to call this part and this part by new names. When they're linked together, we're going to call these residues, amino acid residues. It's what happens after the amino acid comes together to form the amide bond. Now, notice that this is a condensation reaction. Should you be concerned about it being a condensation reaction? Seems a little odd, doesn't it? Why is this problematic in life? Where does this reaction happening? In 55 molar H2O. What about Le Chatelier? How can these things condense in the presence of 55 molar H2O? Isn't H2O going to drive the reaction the other way? So where does the peptide bond happen? Does it happen in solution? Where does it happen? On the ribosome, of course. It doesn't happen free in solution. Okay? What happens to proteins that sit around long enough in H2O? What do you think in 55 molar H2O the equilibrium state is? Is it a protein or is it free amino acids? Free amino acids. So how come you're not all dissolving in front of me? That's the difference between kinetics and equilibrium. And we'll talk about that when we talk about enzyme kinetics. If you leave something long enough, like dead bodies, what do? They turn into free amino acids. Okay. Now, the next hero of 705 happens to be this guy. Anybody recognize him? Greatest chemist in my mind of all time. Linus Pauling, man, Linus Pauling, greatest chemist ever. What is it that Linus Pauling did? What are some of the things he did? Well, he worked on the geometry of peptide bonds. Okay. And in those days, he did it by X-ray crystallography, a fantastic technique that we'll talk about at length. And he measured bond distances and bond angles. What you're looking at here, right, is the crystal structure of a dipeptide. Here's one amino acid. Here's the nitrogen. Here's the alpha carbon. Here's the R group. Here's the carbonyl. Here's the amide bond to the NH group. Here's the next alpha carbon. Here's its R group. Here's the carbonyl. And he measured. You know, one of the things you can do with X-ray crystallography is make very accurate measurements. And he noticed a couple of interesting things here. Well, he noticed, for example, that a typical carbon-nitrogen bond, this was known at the time, is somewhere on the order of about 1.45 to 1.49 angstroms. Right? That's this distance. You can see it's right here between carbon and nitrogen. Okay? And he knew that a carbon-nitrogen double bond, because that had already, those structures had been solved, that was on the order of about 1.27 angstroms. And he noticed something really interesting, which was that this bond, this amide bond, was 1.32 angstroms. It was somewhere midway between a single bond and a double bond. And why is that? Now it's the answer you gave me earlier. Why is that? That's resonance, right? Because, of course, what you have is this situation. You have the situation where the free electron pair on the nitrogen in the amide bond can give rise to this alternative structure. So this is resonance. Now, there's some important consequences of that. And they happen to do with the hybridization state of the nitrogen and the carbon. In order for this resonance form to exist, this carbon and this nitrogen have to both have partial double bond character. So, in order for that to be the case, what's the hybridization state? What's the orbital hybridization state of the carbon and the nitrogen? I heard it, sp2. These must be sp2 hybridized in order for that to occur. If they're sp2 hybridized, then that means I can draw something like this. I can draw carbon, nitrogen. This carbon's going to be bonded to an oxygen. Okay, and these are sp2 hybrids. And so I can draw the orbital off this carbon and the orbital off of this nitrogen, and I can even draw the orbital off of this oxygen. And these will all be shared, right, through two giant pi bonds. Now, the only way that can happen is if there's limited rotation around this bond. Limited rotation. Because if I start to twist this bond and move this angle, then those orbitals are no longer overlapping with the orbitals on the carbon. Okay? And as a result, this carbon, oxygen, nitrogen, hydrogen unit must be planar. And that's what's shown in that box there. It is a planar unit. You will see more of this when we talk about this a little bit later. Now, 2 points to make. First, this CONH bond, there are two ways that I could have drawn this bond, right? I could draw it in cis, or I could draw it in trans. Now, am I showing it here in cis or trans? Raise your hand if it's in cis. Raise your hand if it's in trans. Of course, it's in the trans configuration. And I'm showing to you, in general, the trans configuration and the cis configuration. And you can see exactly what the problem with the cis configuration is. Let me show it to you in one other way. This is pi mole that you will have a chance to use yourself. Build residue. We'll make an alanine leucine dipeptide. Leucine we'll say show sticks. Okay, so here's our trans peptide bond. Here's our carbonyl, here's our NH group, here's our alanine side chain, here's our leucine side chain. Notice how far apart they are. This is pointing in one direction, that's pointing in the other. You can see, right, that if I flip this bond, if I make this bond in cis, I'm going to pull this side chain very close to this side chain. It's going to be overlapping with it. And that's the problem. And that's what I'm trying to illustrate here, where you see that the R groups, which are far apart in the trans bond, are very close in the cis bond. And you can get a flavor for that here. So in general, most peptide bonds that we talk about in this course, by and large, are in the trans configuration. So this bond is usually in the trans configuration. There's one example where it's not so bad to be in the cis configuration, and that's if the bond involves an X-proline bond. So if I have X amino acid bound to proline, as I will show you shortly, because the side chain of proline comes back around on itself, there's not that much difference between the cis configuration and the trans configuration. So bonds with proline sometimes are not in the trans configuration. Sometimes they're in the cis configuration. Proteins are very, very different from each other. Here you see a list of proteins. You can see here's a protein that has about 100 amino acids or so. Here's a protein that has 10,000 amino acids. And here's the biggest protein we know of, called Titin. It has 34,000 amino acids and a molecular weight of 3 million. Okay, it's gigantic. Why is it that proteins have different shapes and activities and different functions? At the top is CD4. That's the protein on T cells that acts as a receptor for HIV, and it also plays a critical role in differentiation. And I'm showing you below the structure of RAS, a molecule that you've all undoubtedly heard about as an oncogene that's involved in causing cancer. And we'll talk about both of these molecules in more detail later in the course. But to understand why they look different and why they function differently, well, of course you know it's because it's due to the sequence of amino acids that compose those proteins. And of course, as we talked about, everything is based on 20 amino acids. And so the fact that we can take 20 amino acids and make proteins that do such a diverse range of things means that those 20 amino acids are a pretty cool set of Lego bricks, right? That we can use to make all kinds of amazing molecules. And to understand this, we have to understand how we might go about this, we have to understand the different properties of each of the amino acids. And of course, each amino acid has a unique character and a unique personality. And when you know this and recognize this, then believe it or not, at the end of today's lecture, and certainly at the end of the problem set, you will be able to look at the sequence of a protein by eye and already infer, based on that sequence of amino acids, some clues about what its biological behavior is likely to be. And this is because the amino acids differ in size and shape and charge, in chemical reactivity and in hydrogen bonding ability. And each amino acid is distinguished by a 3-letter code and by a single-letter abbreviation. And one of the few things that I do actually ask that you memorize in this course is the sequence and abbreviation and structure of all of the amino acids. So, for example, the amino acid glycine goes by the 3-letter code GLY and the single-letter abbreviation G. And each amino acid can be classified into a group based on the chemistry of its side chain. And I'll tell you what they are, and then we're going to fill them in as we go. But to tell you what's coming, we have hydrocarbon side chains, alcohol-containing side chains, aromatics, anions, amides, and cations. I hope I said sulfur-containing. And what we'll do is we'll go through and we'll write each of the amino acids in 3-letter code and 1-letter code, and we'll draw its structure, and we'll talk a little bit about some of the groups. And to try to help you with this, I've given you a very silly little poem, right? I hope some of you took some time to read it. It's called Amino Acid Tales. It's a very amusing way to help you learn the structures of the amino acids. And we'll start first with the first group of side chains, because we're going to break these up, and these are the R groups that I mentioned to you. And the first group, of course, are simply hydrocarbons. And I'll start by showing you for the first or second amino acid the general structure. Again, I'm drawing them in the Zwitter ionic form because we all agree that that's what amino acids are going to look like. And glycine is the easiest of all of them. Gly, G, easy. Something special about this alpha carbon compared to every other alpha carbon of an amino acid of the 20. What's special about this one? Yeah, it's achiral. It's not chiral, right? Because it's got two of the same substituents. Okay? I'll draw one more like this, and then I'm going to stop drawing the amino group and carboxyl group, and I'm only going to focus after this then on the alpha carbon and below. And if this is a methyl group, this is called alanine, ALA. It goes by the single letter code A. Okay, and I'm just going to draw for you now what the side chains are. Okay. So if everyone's okay, I'm just going to draw the alpha carbon like this. I'm not going to draw the amino and carboxyl groups anymore. Okay, and I'm only going to draw now the side chain. This one looks like the upside down letter V. This is that side chain, and this is called valine, single letter code V. Nothing tricky so far. Okay. I can do the same thing. And now I can do the exact same thing I did before. The only difference now being that I have extended that letter V by one methylene group, and this is called leucine, L-E-U, single letter code L. Now I can draw an isomer of leucine that looks like this. Okay. Again, I'd like you to notice that the side chain here contains the exact same number of carbons, 4, as the leucine side chain, but it's an isomer of leucine, and so we'll call it isoleucine, I-L-E, or single letter I. Now, there's something a little curious about isoleucine. What's special about this carbon? Yeah, it's chiral. It's also chiral. So I have two chiral centers, the alpha carbon and this. Okay. There's one more hydrocarbon-based side chain that I need to tell you about, and it's a little bit of a funny amino acid, and we alluded to it already the last time. And that's because this time I'm going to have to draw it a little funny. So here's the nitrogen, hydrogen. Here's my alpha carbon, COO minus, NH. Yeah, 2 plus. And that's because this side chain comes around. This side chain isn't free-floating. It's 3 carbons that rejoins this nitrogen here. And so, in fact, even though I told you that there were 20 amino acids, I lied. Because this is really an imino acid. Although we will forever after refer to it incorrectly as an amino acid. And this is, of course, the amino acid proline, which goes by the single letter code P. Okay, and the side chain for the proline, again, is these 3 carbons. This is the one, remember, that I alluded to was more likely to form a cis-proline bond. Now remember, it's still going to primarily form trans-peptide bonds. But if any amino acid has a tendency to form cis-peptide bonds, it's the prolines. Okay.